Thank you very much. So thank you to Samir and Deb, as well as Leah and Lana, for the invitation to speak to you today about our work and specifically using adaptive focused acoustics to look at macrophages. And we started this project because we really wanted to just try it out and see what we find. And the biological implications that come up in the end were quite exciting to see. So for my lab, we study fungal diseases, and primarily medically relevant fungal diseases, but also those that impact agriculture. And fungal pathogens themselves for humans cause close to 4 million deaths per year. Mortality rates are what really are staggering for fungal pathogens. And so you're looking at 90%, upwards of that even, depending on the pathogen and the location that it's occurring. And some of the reasons for this are inadequate surveillance and diagnostic programs. So in our everyday lives, we're exposed to fungi from the air we breathe to the food that we eat. And so we may have a different level depending on where you live and what you consume, and that can impact your diagnostics, whether you're acute or chronic infection. So they present a dynamic that is quite important for us to study. There's also geographical spreading. You've probably heard the fungal pathogens that move globally, but then also pathogens that were originally plant pathogens that are now able to infect humans. The rising incidences and severity and comorbidities that occur, we saw this quite a bit during the pandemic, where different fungal pathogens, because they're often opportunistic, were able to take hold of hosts, and limited antifungal agents, which we are working to increase our number of agents that are available, but we also see rising rates of resistance. And so my lab has defined 4 pillars then for how we tackle fungal disease research. The first pillar is looking at prevention, and this is where we work with antivirulence compounds to work to disarm the pathogen. And we're primarily looking at natural sources of these, such as mollusks and snails from the environment. The second pillar is diagnostics. Here we're looking to, can we improve diagnostic measures? Can we improve prognostics for fungal infections to really understand what is happening within these situations? And that takes us on to pillar 3, where we're working with monitoring. This is host-pathogen interactions. We want to understand how the pathogen overcomes the host defenses and how the host is trying to defend itself from the fungus. And then pillar 4 is treatment, where we are looking to identify new drugs, new therapeutic targets, and also testing out how we can overcome antifungal resistance. And so with these contexts in mind then, we partnered with Guelph so that we could use our standard proteomics workflow and we could see how it compares. And we also wanted to see if there are improvements that we can make. So working with mammalian cells as well as fungal cells, we have quite a robust workflow that we use. We start with probe sonication to extract our proteins, and then we do a bottom-up approach for discovery-based proteomics. Get our peptides, we run them on the mass spectrometer, and then we process our data using in-house as well as commercially developed programs. And so 2 of the areas that we talked to with Samir and Leah and Deb were very much about where can we improve. So extraction was one of those points, as well as digestion. And we wanted to try this out and see the areas. We looked at tackling it across 3 different instruments. And so we tested the E220 instrument, which is the single sample AFA technology. Then we tested 2 multiplexed AFA technologies that we could process our samples simultaneously. So the LE220 RSC and the R230. So across these 3 instruments then, we set up our experiments. In the first experiment, this is our single sample AFA, we did a direct comparison to our probe sonication. So we have the probe sonicator, we have the E220, I'll refer to it as the E instrument, that we used within this workflow for our extraction. For the multiplex sample, we compared our probe sonication and then between the LE instrument and the R instrument and went through our protein extraction. And then lastly, we also want to test out the difference with lysis and digestion. And so this is where we use those multiplexed instruments. We use them for the lysis stage, and then we kept our samples with Guelph, and they also did a digestion using these instruments too, so that we could see about optimizing those conditions. So for the workflow itself then, if we look at the time, this is our standard proteomics workflow when we're working with very discovery-based, again, these fungi, the mammalian cells are quite difficult. For these particular samples, we were working just with mammalian cells because we wanted to see what is the baseline of what we can detect. And so our standard workflow from start to finish, from collection, lysing the cells, reducing alkylation, and we also do an SDS removal by acetone precipitation for these, although we now are incorporating other methods. Quantification and digestion, purification, and then we measure the samples. And so it was taking about 40 hours in total for our samples to go through this pipeline. And that for the single sample ones, it's the lysis stage would stay fairly consistent. So then, of course, when we move to our multiplex samples for the extraction point, we see a huge reduction in that lysing step. In the overall workflow, it did not decrease it too much in this particular model, or this method rather, but the lysis step is where we see that difference. Because now, of course, we can measure up to 90, or we can lyse 96 samples all at once using the Cavaris instruments, and probe sonication, we're still doing them individually at each time. And if we take it one step further then, and we implement the multiplexing instruments at the 2 points, and particularly working with the Cavaris workflow then, it's the lysis stage, the reduction alkylation, the SDS removal stage, the digestion stage is where we can attack our pipeline and decrease our run times. And so there we were all the way down to 4 hours with the R230 compared to our originally 40. So certainly a great opportunity to decrease the overall time of our workflow, and as well as our hands-on time. So the probe sonicator, you can't put a sample on and walk away. You stand there and you hold it and you probe sonicate. My students know this very well. But for the other instruments where we can put them in and leave it, even though maybe they take the same amount of time, might take 5 minutes for our sample, it still means that you can go and do something else. You can prep the next sample. So it sped up our workflow in numerous ways. So then when we want to look at the data, we started to look at it. It's you want to piece this into different parts. So we started with our single sample AFA technology, and we compared it to the probe sonication. So we looked at number of proteins that we identify, and we can see that we had significantly more proteins using the E220. And we're still seeing a good number. We were around 3,200 up to 3,400. And we had a common proteome of 29, just about 3,000 proteins. But we see unique proteins from both methods that we're able to detect. And it's this uniqueness that we like to dive into further to understand, are there biological implications here? So we then look at the quality of the data, so our principal component analysis. One thing that stands out here, it's not surprising, of course, that they separate on the two axes, in the sense along the x-axis with E220 being on one side and our probe sonication on the other. They're different approaches. This is a big difference in our data set. But what you can clearly see is the distribution of replicates. And so the E220, very, very close replicate reproducibility, whereas our probe sonication, again, this is technical variability that can occur when you process each sample, is evident within this look. If we then do a column correlation, so we quantify the replicate reproducibility, we also see that we have a significant increase using the AFA technology instrument. So that then allows us to look at, if we move beyond the column correlations, and we want to look into our dynamic range of our proteins. And this is where we really started to see the story coming together, because we have this lower dynamic range, as we were walking through the data, is where we really start to see that we're getting more proteins in this lower dynamic range with the AFA technology than we are with our probe sonication. This isn't something that I expected or something that we've seen before or we're working towards, but it was a neat observation. So we kept that in mind. We then went through the multiplex instruments and we tried both of those. So this comparison is with the LE220 RSC instrument and the R230. And again, you see the stepwise increase as we use the different instruments. These are multiplex, so we can run these. We processed 4 to 8 samples at a time, but you can do up to 96 samples. And we can see that increase in protein identifications. Again, our core proteome stays pretty solid at 2,600 proteins out of a total of 3,500 or so. And we have those unique features. And actually, the unique ones here with the two Cavaris instruments was an 18% increase in protein IDs that we hadn't seen using our probe sonication. So again, we look at our principal component analysis and we can see our separation here across the different sample sets. Fairly similar, I would say, in their distribution. We had the R230 here has a little bit more distribution here, which we think might be from our samples themselves. We looked at our column correlation, and the PCA translates nicely over there. So we've got our increase in proteins that we see and the replicate reproducibility as well. And then if we look at our dynamic range across the samples, again, we see with these Cavaris treated samples or the Cavaris instrument samples that we have these lower abundant proteins. This dynamic range is a bit extended using this. So these are observations, and that's strictly from the lysis and the extraction. So then we moved into that combination of lysis and digestion. So not only did we decrease our workflow timeline, but here we also wanted to see what else could we get from it. Are we missing anything? We typically do our digestion at 30 to 37 degrees and without agitation, and so this does add another layer of complexity. So here we saw an increase in our protein IDs when we compared it to our probe sonication approach. We have our typical core proteome. We do have unique proteins, again, that we're identifying. We also have some unique proteins in our probe sonication, so we do keep those in mind, and we look at those again later too. And we profiled out from our PCA plot here the separation of the instruments. So the multiplex instruments cluster really nicely together, which we would expect, and then our probe sonication. And again, there's still some replicate reproducibility, a bit of distribution across our replicates. It's fairly constant, but we did see across all of our samples increase in protein identifications, increase in replicate reproducibility, and an increase in our dynamic range when we use the AFA technology. And again, with our dynamic range here, we saw very clearly on these ones when we combined extraction and digestion, those lower abundant proteins. So the next thing that we want to do, and I think the most exciting part for us then, is to start to look into what are these exclusive proteins that we are detecting using the AFA technology, and especially what are the low abundance proteins, because are those something that we are missing in our other approaches? So when we look at the proteins themselves, this is where we see we focused on, this analysis is on the AFA exclusive proteins. So we had over 1,000 proteins that were identified across the different AFA technologies that were not identified in probe sonication. And then we focused even further, because I wanted to be quite conservative in our analysis, and we looked at only the proteins that were identified in all of the AFA technologies. So not 4 out of 5 approaches, not 3 out of 4, we had to have it in all 5 different methods, and absent completely in our probe sonication. And so those are the ones that are highlighted in red there, and we had 86 proteins. So we did narrow it down quite a bit, and I wanted to see if we push this analysis, what do we see in these particular proteins? And so in that approach then, what we noticed, we looked at the dynamic range, so the LFQ, our label-free quantification intensities, because I wanted to see, we noticed a dynamic range on the S-curve, but does it actually matter? And the median LFQ intensity does have a significant decrease, so we are able to get lower abundant proteins using the AFA when we look at those AFA exclusive identifications. So if we take those 86 then and we put them into a string interaction analysis to see if there is something relevant there. And here's where we found the interesting side of things. Because we're working with macrophage, and we do a lot of host-pathogen interaction work in my lab, immune signaling and inflammatory responses, transport, those are all things that we characteristically look for. So within the macrophage, even in an uninfected situation, we see that we have inflammatory response being activated. We have cytokinesis that's being turned on. We had different transport. We had endocytic transport. We also had exocytosis. And so these different biologically relevant areas that we look at anyway when we look at our infection settings, that came out. And then we also looked a little bit further. We looked at different gene ontologies, whether it be biological processes, cellular compartment, molecular function. We looked at KEGG pathways, and we noticed similar patterns in our data set where we had these vesicle-mediated transport proteins that are coming out. We had different metabolic processes. But again, this transport and immune response was something that we observed across the board. And so that was quite interesting for us because it tells us that these are something that we are missing in our current workflow with probe sonication, likely because of heat that is added to the sample. Even if we do it in an ice bath, there is still that difference. We did the same analysis with the proteins that were identified only in probe sonication, so the exclusive to probe sonication. And there we found ribosomal proteins, a few translational proteins. It was very scattered. We only had one cluster that came up with 3 proteins, and the rest were all individual. So we are seeing something new here that we hadn't expected from our data. And so what we took from this then is what did we find within this study? We have improved workflow efficiency as well as efficacy. So we had better protein identifications, a higher dynamic range, as well as replicate reproducibility was higher as well. We had robust proteome coverage across the board. So no matter which method we used, we identified fairly similar numbers of proteins, around 3,500. I should mention that this was also data-dependent acquisition, and this was done on a 240 originally. We've done some later studies with the Astral Zoom. And so the instrument itself, as well as the DDA approach, can also be something that we're working to improve. We have strong quantitative reproducibility and new biological insights that we were missing before with our approach. Some of the considerations then, so we improved alignment and outputs of robust and reproducible results from the probe sonication platform. And we looked at alternative conventional approaches. And so right now when we work with macrophage, we did the probe sonication. This is excessive for macrophage, but when we do our infection studies with the fungus, we need to use that's what we thought. We need to use the probe sonication. So we could also compare this to more of a water bath sonication or other approaches that are perhaps less harsh or less single sample dependent. And also another consideration then is the cost of instrumentation and consumables. This is something that when you're looking at purchasing technology to consider. What I really liked about this project is the future directions. I think this is the most exciting part of it, at least for myself, and I think too, after talking to Samir, is where can we take this? So now that we've seen we can identify these proteins, these low abundant host proteins, and we want to study an infection setting, where can we take it and what can we do? So we are working to further explore those AFA exclusive proteins. There's about 1,000 of them, so we're looking at categorizing those, our string interaction maps, as well as network mapping in different modalities, to modify the probe sonication workflow for reduced timelines. And so we can move whether we use water bath sonication, probe sonication, alternative SDS removal approaches, and then of course the downstream testing, moving into data independent acquisition, because I think we would see even more dynamic range with our samples in that sense. Well, I know we would see more, we've done that part. Incorporate updated digestion guidelines based on in-lab testing, so this is something also that we can work to do. Improve mass spectrometry instrumentation and acquisition methods, and so that will push the identifications further. And I also think that our replicate reproducibility would be even higher, which then ties into our replicate samples, our imputation levels, all those come into play when we start to work in that space. And then our infection applications and profiling. So as I mentioned, this is macrophage only, but our next step then is to set up our infection settings where we work Cryptococcus neoformans, the fungal pathogen. We can put that on the macrophage, and then we can really start to see what are the inflammatory associated immune response proteins that we've missed before. We also see that with new mass spectrometry technology, our detection of fungal proteins in an infection setting has increased dramatically. And so we can also, I'm excited to see if we also detect more of our fungal proteins using this approach as well. And so with that, I want to thank my lab members that assisted and worked on this project and prepped the samples. I also want to thank the team at Kavaris. So Samir and Leah that processed all of our samples and really made this possible and help us set up a nice, efficient experimental workflow. We do have a poster today. Leah will be presenting the poster, and so I invite you to check that out. And then there's also a preprint that we've put up on bioRxiv, and so you're welcome to check that out as well. Thank you very much.